Welcome to my YouTube channel. Today we are going to talk about the genomic fingerprint, B cell and T cell clonality and the basics. Very basic pretest.
How do you define a clone? So clone can be defined as a population of lymphocytes carrying a unique signature. So here you can see that diverse lymphoid populations arising from multiple progenitors are seen. They carry different molecular fingerprints. This is typically seen in reactive and inflammatory conditions, whereas massive expansion from a single progenitor cell carrying a unique fingerprint, one dominant fingerprint, is suggestive of lymphoma or leukemia. So polyclonal population gives you a classical Gaussian distribution on PCR, whereas a single dominant clone as seen in malignancies will give you a sharp spike like this. So why do we do clonality testing? Clonality testing, as we have just seen, it helps to distinguish neoplastic lymphoid proliferations from reactive processes. It identifies a monoclonal population which can indicate a potential lymphoma or leukemia. And clonality testing can also be used for monitoring your residual disease. How do you define immunity, and how many types of immunity do we have? So in simple terms, immunity can be defined as a defense against microbes. So there are two types of immunity: innate immunity and adaptive immunity. Innate immunity is something which is already present in the body. Adaptive immunity is created in response to exposure to some foreign substance. Innate immunity is non-specific. It can fight any foreign invader, whereas adaptive immunity is specific. It can only fight against specific infections. Innate immunity is rapid, adaptive immunity is slow. And innate immunity has got limited and lower potency, whereas adaptive immunity is highly potent. Once activated, innate immunity remains throughout the life, whereas adaptive immunity, it can be there for lifelong or for shorter duration. What are immunoglobulins? How do you define immunoglobulins? What is the structure of immunoglobulin? So immunoglobulins are nothing but glycoproteins. They consist of two light chains and two heavy chains. There are different types of heavy chains that are there, based on which we have 5 subclasses of immunoglobulins. Gamma stands for IgM, alpha stands for IgA, mu heavy chain will form IgM, epsilon heavy chain will form IgE, delta heavy chain will form IgD. So there are two types of light chains called kappa and lambda. If you see the immunoglobulin structure, as I said, there are two light chains represented in blue here, and the yellow ones are showing your high chains, heavy chains. So the blue color represents light chains and the yellow color represents heavy chains here. This is antigen binding site. This is FC region, fragment crystallizable. This region constitutes our antigen binding site. This is hypervariable region, this is variable region, and this is constant region. Light chains and heavy chains, they are linked by disulfide bonds. If you see the structure of immunoglobulins, IgA is a dimer, IgM is a pentamer, remaining 3 are monomers. You can see that the heavy chain, based on heavy chain, the immunoglobulins are named accordingly. The mnemonic is GAMED, you can remember GAMED. Now we know that structure of immunoglobulins and composition of immunoglobulins, different types of immunoglobulins. So let's see what are T cell receptors. Any idea? We'll discuss in subsequent slides. What is VDJ recombination? Have you heard of it? So VDJ recombination in simple terms, recombination of V segment, D segment and J segment of immunoglobulin and T cell receptor, which is essential for differentiation of B and T cells. We'll see VDJ recombination in detail in subsequent slides. The difference between IgH and TCR-g assay, and why we are using IGH and TCR-g as the most common test for clonality. Let's find the answers in subsequent slides. Have you heard of recombination signal sequence? Recombination signal sequence is a non-coding DNA sequence that can be found next to the points where recombination usually occurs. They act as signals for the recombination process. That's why they call recombination signal sequence. So because of recombination process, we see lot of shuffling of gene segments. RSS are located at 3' end of V gene segment, 5' end of J segment, and on either side of D segment. This is the location of RSS. So now let's begin our class from the basics. You know that from hematopoietic stem cell we have common myeloid progenitor cell and common lymphoid progenitor cell. From common lymphoid progenitor cell we have T cell, B cell and NK cell. B cell in turn differentiates into plasma cell. Common myeloid progenitor cell gives rise to other cell lineages like megakaryocytes, granulocytes, erythrocytes, etc. So this is just to show you that T and B cells constitute adaptive immunity and the remaining cells constitute innate immunity. If you think that this white portion of the slide is your body, then the blue color here represents immune system. You know that immune system has two types of immunities like adaptive immunity and innate immunity. So adaptome can be defined as all adaptive receptors including Ig and TCR receptors that an individual possesses is called adaptome. So this is an individual makeup, adaptome, whereas repertoire is a subset of adaptome. At any given point, the total number of B cell receptors and T cell receptors will constitute immune repertoire. So if I take your sample today and try to see like how many Ig receptors and T cell receptors you have got at specific time point, then that constitutes your immune repertoire. So in simple terms, all the clonotypes will constitute your repertoire, immune repertoire. Then what is a clonotype? Clonotype is a functional unit of immune repertoire. It carries a unique CDR3 sequence. Remember this thing, unique CDR3 sequence. Each clonotype is defined by its unique CDR3 sequence. So now we are interested in studying clonotypes only. There are other assays which can study whole immune repertoire by repertoire sequencing, but with clonality we are very much interested to know the clonotype of an individual to differentiate the clonal population from polyclonal population based on the clonotype signature, to be specific CDR3 signature. As a human, we have a fundamental challenge that our genome is very small to encode a specific receptor for every pathogen that we are going to encounter. To solve this, nature has given a phenomenon called combinatorial diversity, which is created at the DNA level. So as a result of which, a unique heritable molecular fingerprint for every lymphocyte is being generated. So this is a very small example, visual representation to show you like we have finite germline genes which is getting recombined with the help of RSS. Just now we have seen that. So because of the recombination, it is able to generate enormous amount of receptors or immunoglobulins, generating antibody diversity. So this biological paradox of generating 10^12 different immune receptors with the static genome is called a combinatorial diversity. If you take a simple example, in simpler terms, you can just think of lock and key mechanism here. VDJ recombination to generate diversity, it is creating hundreds and thousands of different types of keys. So when the right key fits the right lock, the body will try to give signals to the cell to expand rapidly so that it can make this one key into hundreds and thousands of copies, so that you can effectively fight against your infections. In the year 1987, Susumu Tonegawa, he for the first time discovered the genetic principle for generation of antibody diversity. This discovery has challenged the fundamental concept that one gene encodes one polypeptide chain. Usually one gene encodes one polypeptide chain, that's what we know, but in case of B cells and T cells there is lot of diversity which is being generated to combat our infections. If you see B cell development, there are two phases basically: antigen independent development which happens in bone marrow, antigen dependent development which happens in secondary lymphoid organs like spleen and lymph node. We have seen that hematopoietic stem cell gives rise to some common lymphoid progenitor which forms precursor B cells. So these precursor B cells, they acquire VDJ recombination and they become naive B cells. Once they successfully attain this VDJ recombination and are able to express IgM on the surface, they will be released into circulation. And these naive B cells now, they look for antigens. If they don't encounter any antigen in a given span of time, they undergo apoptosis. If at all, if they encounter any antigen, so further activation happens in the secondary lymphoid organs like spleen and lymph node, where they undergo somatic hypermutation and class switch recombination. Hematopoietic stem cell giving rise to common lymphoid progenitor, and which by acquiring heavy chain gene rearrangement partially becomes a pro B cell. Pro B cell, after attaining a complete heavy chain gene rearrangement, will become a pre B cell. Pre B cell now acquires light chain gene rearrangement to become an immature B cell. After some RNA processing, the immature B cell becomes mature B cell. This mature B cell, after antigen stimulation, becomes an activated B cell, which differentiates into IgM secreting plasma cells. And class switch recombination can also happen based on the requirement. Initially we have only IgM and IgD on the surface of the B cell. So based on the requirement, the class switch recombination happens and it can differentiate into IgG, IgA, IgA. So let us now discuss about somatic hypermutation. Here I said that naive B cell, after encountering some specific antigen, it undergoes further clonal expansion and somatic hypermutation. So the spontaneous mutation rate allowed for a normal cell per cell division is around 10 power 8 to 10 power 10. That means one mistake is allowed out of 10 billion base pairs that are getting generated per cell division. But somatic hypermutation will make one mistake in 1000 base pairs. Like for every 1000 base pairs that are being generated after each cell division, one mistake would be there. So this cell division, as it goes on, the mutations can accumulate, and this phenomenon is called somatic hypermutation. This is mutagenic process, but this is very important for the affinity generation. So the B cells with improved affinity can bind to antigen and they can eliminate the antigen successfully. Those B cells with decreased affinity, they undergo apoptosis. After this affinity maturation, class switch recombination can happen based on the requirement. So for both somatic hypermutation and class switch recombination, you need an enzyme called activation induced deaminase. Activation induced deaminase has got two different roles here. In somatic hypermutation, it converts cytosine to uracil. It converts all the cytosines to uracil. Whereas for class switch recombination, it makes changes in the constant region, because constant region is the one which is different among all the immunoglobulin types. So it makes changes in the switch domain of constant region to differentiate one immunoglobulin type into other immunoglobulin type. This is just to show you the sequence of development of B cells. B cell precursor becoming naive B cell. After antigen exposure, it moves to secondary lymphoid organs where it is called a centroblast in the germinal center. Centroblast differentiates into centrocyte. Centrocyte will differentiate into plasmablast, and plasmablast finally differentiates into plasma cell. I just want you to remember one thing here. Chronic lymphocyte leukemia, the precursor cell can be pre-germinal center B cell or post-germinal center B cell. Just remember this, the cell of origin for this. So these are like out of context, but still you can, if you want, you can go through. For germinal center type DLBCL, the cell of origin is centroblast. For follicular lymphoma, this is centrocytes. And for marginal lymphoma, marginal zone lymphoma, this is marginal zone B cell. Plasmablast can give rise to ABC type of DLBCL, and plasma cell, you know, it causes multiple myeloma after acquiring translocations. So again you can see that the very eye-catchy thing here is like 14, chromosome 14, where our IGH lies. So IGH is very notorious. It causes promoter hijacking. Whenever an oncogene like MYC, BCL2, BCL6 come closer to IGH, it will just take over. It takes the control of cell proliferation, differentiation. So that's how we get so many lymphomas involving the IGH locus. Let us have a look at T cell development now. You know that T cell development happens in thymus, and it is regulated by Notch signaling and interleukin 7 receptor signaling. The few initial phases of T cell development are double negative. That means they don't carry CD4 or CD8. So they are again subdivided into DN1, DN2A, 2B, 3A, 3B, 4 based on the cell surface expression. We are not going in detail about cell surface expression, how these differ from each other. We are only focusing on the rearrangements which happen and the order of rearrangement, how does it occur. So after DN2A, the cell will undergo gamma delta rearrangement. Okay, so gamma delta rearrangement, if it is successful, if it is leading to a productive rearrangement, that is a functional protein, then the lineage will be assigned as gamma delta T lymphocyte, and the cell will leave thymus at this stage. So gamma delta T lymphocytes do not carry any marker CD4 or CD8 markers. Okay, they are double negative and they do not undergo this positive selection. This is happening for MHC. So these are MHC independent lymphocytes. They don't need MHC for antigen processing, antigen recognition. If at all, if gamma delta rearrangement fails here, that means if it is leading to non-productive rearrangement or non-functional protein, then the cell has to undergo remaining phases. After DN3A, TCR beta rearrangement happens. So beta rearrangement, if it is successful, the cell will continue to progress through these stages, and it acquires alpha rearrangement after ISP phase. After this phase, TCR alpha rearrangement, if it is successful, then the lineage will become alpha beta. At this junction, TCR alpha and TCR delta, they are located on same chromosome. So whenever TCR alpha rearrangement is happening, if it is leading to productive rearrangement, delta, which is placed inside the TCR alpha genomically, that region will be deleted. So cells will lack TCR-d signature here. But somehow all the cells have undergone gamma rearrangement at the very beginning of the development. So all the T cells which are going through this pathway, they tend to harbor gamma signature, TCR gamma signature, but that will be non-productive. the functional protein and functional lineage will be alpha beta, but they carry this molecular scar which was there in the early development, whereas delta is getting deleted once you once you get alpha rearrangement. So in simple terms, once you get alpha beta lineage, delta is getting deleted, so you cannot test for delta rearrangement, whereas gamma is located on some other chromosome, so it is carried forward along with the different stages of T cell development. That's why we focus and we screen all the T cell lymphomas for gamma, TCR gamma assay. That doesn't mean that all the T cells are gamma delta T lymphocytes, but all the T cells will carry this molecular scar of gamma rearrangement, which can be productive or non-productive. That can only be confirmed by doing TCR beta assay. So TCR beta assay, if it is positive, that means that the cell has got both gamma and beta signature, but the assigned lineage will be beta. If TCR gamma is positive, beta is negative, that means the cells belong to this subset, gamma delta subsets, because gamma delta T lymphocytes has never reached this point to attain that beta rearrangement. Kappa light chain is located on chromosome 2, lambda light chain is located on chromosome 22, heavy chain is located on chromosome 14. If you see the distribution of variable diversity and joining segments across these three, heavy chain, as the name suggests, it has got high molecular weight. It weighs around 50 to 75 kilodaltons, whereas light chain weighs around 25 kilodaltons. Heavy chain has got highest number of variable regions, diversity regions, and joining regions. If you observe keenly, light chain lacks diversity region. Both kappa and lambda, they lack diversity region. There are 4 different types of T cell receptors like alpha, beta, gamma, and delta. Alpha and delta are located on chromosome 14. Beta and gamma are located on chromosome 7. So that's why I was saying that alpha rearrangement, when it happens, it will automatically delete delta locus. That's why we don't see delta rearrangement in alpha beta positive cells. And beta delta, they both have D segments, whereas alpha and gamma, they don't have D segments. So kappa, lambda, alpha and gamma, these chains, they do not carry D segments. We have already seen the structure of immunoglobulin, that it has 2 heavy chains and 2 light chains. T cell receptor is a heterodimer which comprises of alpha chain and beta chain. It's a heterodimer. Majority of the circulating T cells are alpha beta cells. They found in lymphoid tissues. Less than 5% of T cells are like they harbor gamma delta signature. So they constitute less than 5% of T cells. They are present in epithelial tissues. So the basic difference between immunoglobulin and T cell receptor is that immunoglobulins can recognize free unprocessed antigens, whereas TCR alpha beta specifically, it requires antigen presentation via MHC molecule, whereas gamma delta, they are MHC independent. They don't need MHC to identify antigen. Like immunoglobulins, they can also process very different kinds of antigens. They can also recognize lipids, etc. So they are not basically MHC dependent like your alpha beta. Here these are the V variable segments numbered from 1 to n, D segments numbered from 1 to n, J segments numbered from 1 to n, and C segments are represented here. C segment, this is the site where you decide whether you are looking at IgM or IgG, Ig alpha or IgE. So mu stands for IgM, delta stands for D, and epsilon stands for E. So only one of it will be there, not all together, just to show you this is schematic representation. And one more thing, very important thing to remember is that every variable region is preceded by one leader sequence. You can see here LN, L1 to LN. They are also there in the similar numbers. So first rearrangement to be happened in B cell development or T cell development is that of DJ rearrangement. So DJ rearrangement, you can see here D segment, the first DH and here JH, they came and fused together with the help of RAG. We'll see what is RAG in subsequent slides. So the intervening segments are being deleted, only this has come closer to JH. They both got fused. This is called DJ rearrangement. So DJ rearrangement, when it combines with any one of the variable region, it forms VDJ rearrangement. It is called VDJ. This combination is called VDJ. Okay. You can see here that the leader is there before variable. So even transcription, after splicing, all the intervening regions have been deleted. Only the leader, one variable segment, one diversity segment, one joining region is there along with constant region. This is the final product. So the human genome, the germline DNA is same, but because of various combinations that are happening, randomly one variable, one diversity, one joining region will be there as a final product. So each lymphocyte will carry a unique VDJ recombination. Let me show you with one more example. Here it is clearly numbered from 1 to 62. Variable regions are spanning this area. D genes are numbered from 1 to 27. J genes are numbered from 1 to 6. Say suppose from here D2 is coming and getting fused with D5, J5. So you see D2, J5 here. After successful DJ rearrangement, this DJ now fuses with one of the variable region. Say suppose here it is fusing with V3. So like that, so many combinations are possible. So now finally you have got a clonotype with IGHV3, IGHD2, IGHJ5. If you look at the structure of this segment, this clonotype, it comprises of one leader sequence, and the region is subdivided into 4 framework regions: FR1, FR2, FR3, FR4, and the intervening regions are called complementarity-determining regions. So FR1 is followed by CDR1, FR2 is followed by CDR2, FR3 is followed by CDR3, and at the last we have FR4. Now what is very important is that you have to remember CDR3, because it is lying at the junction of V2D and D2J. This is undergoing huge diversity. I'll tell you why. Here these arrows represents the primer designing. The forward primer spanning IGHV region, FR3 region, and IGHJ indicates your reverse primer spanning FR4 region. So if you target here, you will get IGHV, IGHD, IGHJ sequence. If you design primers from here in between CDR3, forward primer spanning CDR3 region, part of CDR3 region, and reverse primer spanning FR4 region here. So it will give you a sequence comprising of IGHD and IGHJ. I'm just showing you an example of alpha and beta chain. So whatever the structure is, like the distribution of variable, joining, and constant regions is, ultimately one variable segment, one J segment is coming and fusing together to give a final product. Here also one variable, one diversity, and one joining, one constant region is giving you the final product. So IGH library generation can be done from genomic DNA as well as from cDNA. If you want to study somatic hypermutation, you better take cDNA as the template and use the primers, forward primers spanning from leader sequence, and reverse primers spanning from this constant region. It will give you a good amplicon, but you need good quality material. From if you're using genomic DNA as the template, then you can use multiple sets of primers like FR1, FR2, FR3, the reverse primer from joining region. The amplicon length will be smaller here. You can very well use this if your quality of DNA is poor. Now comes the recombination signal sequence. We were discussing about RSS. There was a question regarding RSS previously. So this is a short DNA sequence. It's a non-coding DNA, indicates the sites of recombination. If you zoom into RSS, you can see that there is one heptamer on the 5' side that is composed of 7 nucleotides, followed by a spacer which can be 12 base pair long or 23 base pair long, followed by a nonamer which is a sequence of 9 nucleotides. It is there on the 3' side. One more thing is to remember is that 5' heptamer and 3' nonamer, they are conserved nucleotide. That means the sequence remains constant. Whereas 12 base pair, 23 base pair space, it is unconserved. Like it can have variable combinations of nucleotides in between. As we have already seen, each V, D or J gene segment is flanked by a recombination signal sequence. You have to remember 1223 rule. This is a kind of regulation on recombination of VDJ segments. We have seen that RSS can be of two types with 12 base pair spacer and 23 base pair spacer. This is called 1-ton RSS. This is called 2-ton RSS. Okay. So if you have a gene segment with V, for example V segment with 23 base pairs can fuse with D segment with 12 base pair RSS. Okay, 23 base pairs V region cannot bind with 23 base pair RSS flanking D region. That means RSS of same type cannot come closer and they cannot help in recombination. Like 23 base pair RSS and 23 base pair RSS from other gene segment cannot combine together. So one gene segment with 23 base pair can only combine with the other gene segment with 12 base pair. Now let's talk about recombination activation genes. So these are called RAG1 and RAG2. With the help of high mobility proteins like HMG1 and HMG2, they help in recombination process. So RAGs, they can recognize RSS sequence. So we know that RSS sequence is here. This RSS sequence is here. These RAGs, they go and bind to RSS. They act as a molecular scissor. They recognize these 12 and 23 because only 23 and 12 can pair. So they go there and they introduce double-stranded breaks in the DNA. So RAG1 is binding here, RAG2 is binding here. So RAG1 and RAG2 now has to come closer so that this V segment can come closer to DJ segment. Once VDJ comes closer, now this recombinase complex, it can introduce single stranded breaks here to separate this RSS sequence along with the intervening sequence, and VDJ will be separated. Once you get VDJ, the leftover part will be deleted. So to simplify it, RAG1 and RAG2 enzymes, they cleave germline DNA wherever they find RSS. That too 1223 RSS. That rule has to be maintained. Once they cut the double-stranded DNA, they shuffle the segments. First D segment joins with J, then DJ joins with V. This delete something in between. V to D you have deleted something, and D to J also you have deleted something. So you have to now fill the gaps. To fill the gaps, this TDT comes into play, and there is loss of some DNA material here. So we don't know the template. So it will introduce N nucleotides, that means non-template nucleotides. So creating junctional diversity. This VDJ, the fusion, the rearrangement which has happened here, will create a combinatorial diversity. At the junction of these three, when the nucleotides are getting inserted randomly, this will create you a junctional diversity. So there is combinatorial diversity and there is junctional diversity also. Because of these two phenomena, you are able to see a theoretical diversity of 10 power 12 unique combination. Again I am just repeating it. There is RSS here, RAG1 and RAG2. They bind at the RSS. They follow 1223 rule to ensure correct alignment, and they bring RAG1 and RAG2 are there on either side of RSS, and they, when they come closer, this phenomenon is called synapsis, and they cleave the DNA. This leftover DNA will be deleted, and the now unique VDJ will be repaired by non-homologous end joining repair pathway. This VDJ junction region is CDR3 region. That's why I said that each clonotype carries a specific CDR3 signature. So this CDR3 signature will be unique to every clone. Now coming back to somatic hypermutation. Somatic hypermutation, we have seen that after every 1000 base pairs there is a one mutation which is going to happen for every cell division. So because of which you find many mutations in the patient sample. So what you do is you try to compare your patient sample with germline DNA. If you see less than 2% deviation, that means the cell has not undergone somatic hypermutation. So you say this is unmutated, and it carries poorer prognosis because I told you to remember two cells of origin for BCLL. One is pre-germinal center base cell, the other one is post-germinal center base cell. This has not undergone somatic hypermutation, that means this is of pre-germinal center origin. If you see a sequence deviation of more than 2%, that means only 97% of your germline DNA is existing in your patient. The rest 3% or 4%, whatever it is, which it should be more than 2% deviation. So if it is there, you can assume that the cell has undergone somatic hypermutation. It carries better prognosis because the cell has already undergone somatic hypermutation and so much diversity, combinatorial, junctional, and on the top of it, it has also undergone somatic hypermutation. This will behave very less aggressively now. It is not that active now, and so the CLL will be very much indolent here, and these CLLs, they respond well to chemoimmunotherapy also, so they carry better prognosis. On the other hand, when B cell is arising from a pre-germinal center B cell, that is more aggressive type of B cell, which has not undergone so much somatic hypermutation and all, so it will be aggressive, and it will be difficult to eradicate such kind of B cells with chemoimmunotherapy. So the response is poor. So overall it carries poorer prognosis. We have seen that somatic hypermutation is good in the way that it increases affinity maturation. Now we have already seen the prognostic implications of somatic hypermutation in cases of CLL. But in such cases you find difficulty because your primers may not bind to the sequence because of the mutations which are spanning in the variable region. So what you have to do is you have to design primers in such a way that you can bypass the somatic hypermutation signature which is there in the variable region. So what you do is, I told you to remember leader sequence also. Before every variable region, leader sequence was there. So you target that region, you design primers against a leader sequence, so you can skip this variable region and you can get a better amplicon to look for clonality in these cases. Apart from using the framework and leader primers together. There are other primer strategies like degenerate primer sets, 5' race based approaches, and constant region primers, which are largely unaffected by somatic hypermutation, because somatic hypermutation will not affect the constant region. So you can design primers against constant region to amplify your DNA. We have seen the VDJ recombination and the primer design and all. What if IGH comes out to be negative somehow? Otherwise, if you know that IGH rearrangement is there, you want to know the kappa or lambda restriction. So in such cases, you have to have primers for IG kappa rearrangement also. For that, you need to know the structure of IG kappa. I told you that light chains like kappa and lambda, they lack diversity region. So you have variable regions and joining regions. Only one variable region can combine with one joining region. The concept is same for everything. So if you want to design primers, you have to design a forward primer spanning VK and the reverse primer spanning JK. Until now this is looking good. If you design two primers, this is very straightforward thing, like you don't have much complexity here, so you can get your amplification very easily. Successful amplification can happen without allele dropout, etc. But the problem comes when you don't have IG kappa also, because as you know, majority of the B cells they express kappa, but around 30 to 40% of B cells express lambda. So whenever there is no kappa rearrangement, the cell has to undergo lambda rearrangement to survive. Before that, there is some checkpoint which we have to understand. So this is the kappa light chain gene rearrangement. You see that variable segments are there, joining regions are there, constant regions are there. But on the 3' end, you have seen you are seeing something called KDE. KDE stands for kappa deleting element. And in between you have some intron enhancers and RSS also. You know that every VDJ should be flanked by RSS. All this we have read. Okay. So when you have VK and JK, when you have this thing kappa light chain on the B cell, when the B cell wants to express kappa light chain, you get VJ recombination. So you get VK JK as we have seen in the earlier picture. Suppose say that the kappa light chain is giving some non-productive rearrangement. It is not leading to a functional protein, so cell will not express IG kappa. In that case, the cell has to move on. First, allele 1 will undergo IG kappa rearrangement, followed by allele 2. Both the alleles, if they lead to non-functional rearrangement, that is non-productive IG kappa, so that cannot be expressed on your cell. So cell has to go for lambda rearrangement. Before going for lambda, you have to erase kappa signature from here. You don't want kappa signature to be there, and you don't want the cell to go for lambda rearrangement, because when I try to design a primer for IG kappa, that non-reproductive rearrangement will be picked up by my primers. So I won't be knowing whether that is functional rearrangement or non-functional rearrangement until unless I design I take a template from cDNA instead of taking the template from genomic DNA. That is the only way to differentiate functional and non-functional rearrangements. But usually we don't do cDNA, we don't take cDNA, we take genomic DNA. So the body has a beautiful mechanism of deleting non-functional alleles. For that, we have got KDE, that is kappa deleting element. So there are two types of rearrangements which can happen if the allele is non-functional. One way of deleting this IG kappa segment and bringing VK and KDE together. This is one way of doing it. So now this leads to VK KDE rearrangement. In between everything, the intervening sequence has been deleted. The other way of doing it is you delete the prior portion, the variable region portion, there is JK, CK and intronic region with RSS which will fuse with KDE. This is a longer rearrangement. This can also happen. There are two types of deletions that can happen. So whenever there is no Ig kappa, you have to use these two as surrogates to know that Ig kappa has got deleted and somehow the cell has undergone Ig lambda rearrangement. So to use the surrogates, we should know this mechanism. So here you can see, if you want to know whether VK-KDE rearrangement is there, you will design a forward primer spanning VK, reverse primer spanning KDE. Similarly, forward primer spanning JK-CK, reverse primer spanning KDE rearrangement will give you an answer whether this is there or not there. So now whatever I've told you, I'll just try to show you in this image, and you must be aware of the concept of allelic exclusion also. So as I told you, at the pro B cell level we have IGH rearrangement which is there in all the cells, in all the B cells. Here also IGH rearrangement, first it begins in one allele only. Allele 1, if it undergoes VDJ recombination, if it is leading to some functional rearrangement, then the pro B cell can become pre B cell. Okay. If this allele 1 which has tried undergoing VDJ recombination has led to some non-functional protein or unproductive rearrangement, then allele 2 comes in place. So allele exclusion phenomenon explains that only one allele at a time can show you functional rearrangement. If one allele is showing functional rearrangement, there is no chance given for this allele 2 to undergo functional rearrangement. Okay, only when it fails, the body allows you to take allele 2 for VDJ recombination. If it becomes functional, then the cell will become pre-B cell. Again, if it becomes non-functional, if you don't have IGH rearrangement only, because both the alleles were leading to non-functional rearrangement, the cell has to undergo apoptosis. This will not survive, this cannot survive. Okay, either one of the alleles should be functional. Similarly, for Ig kappa also, allele 1 rearrangement will happen if it is leading to successful productive rearrangement. It will be expressed on the B cell receptor. If it is leading to non-functional rearrangement as this, the allele 2 can give a try for Ig kappa rearrangement. If allele 2 is also non-functional, here also you have one step where it undergoes apoptosis. It is not shown here, just to make it simpler. If both the alleles fail to show non-functional rearrangement, you have to erase the signature, non-productive rearrangement you have to delete. For that you have KDE rearrangement and SINT. SINT is that CKGK intron RSS rearrangement. Those things happen. Along with that, cell will undergo Ig lambda rearrangement, and those cells with Ig lambda rearrangement, they express lambda on the cell, on the B cell receptor. I think this is clear now. Once again, I am just repeating it. Initially, cell has to undergo IGH rearrangement. So, we are using IGH as screening test because all the cells will undergo IGH rearrangement no matter the type of immunoglobulin, because the type of immunoglobulin is decided by constant region, not by variable region. So, all the immunoglobulins will have this rearrangement. Then the Ig kappa rearrangement should happen. If it is non-functional, then Ig kappa deletion happens with the help of KDE. It is leading to some gene rearrangement that can also be used as a diagnostic marker because it rearranges V genes to intron RSS to eliminate non-functional Ig kappa alleles. So finally you see lambda chain rearrangement. This is just a repetitive. This is the universal rearrangement which is there in almost all B cells. So we are using, and even because of its maximum diversity, we are using IGH as a unique fingerprint in case of B cell lymphomas. So this is the gold standard screening test. That's it. So primary screening test is IGH and TCR-g. What we are doing, if it is negative or inconclusive, we can very well go for Ig kappa or TCR-b. Ig kappa is crucial when you suspect somatic hypermutation. And TCR beta, it has got higher specificity. To resolve unequivocal cases, equivocal cases, you can use TCR gamma. Why we are looking at TCRG? Even though they constitute less than 5% of your T cells, the molecular star which is there in the early development is still there, so there is no mechanism to delete it. That's why we are able to do a screening test to demonstrate TCRG. So irrespective of the cell type, whether it is gamma, delta or alpha, beta, you have TCRG rearrangement, because this is lineage independent marker, occurs early in ontogeny, covering both the lineages. Even if you look at the structure, it lacks D segment. So it will reduce the PCR dropout rates. It is highly sensitive but less specific when compared to TCR beta. So the proposed clinical algorithm based on our understanding now is to screen IGS with the help of FR1, FR2, FR3 primers. It can capture clonality in almost 90% of the cases. If there is somatic hypermutation, you take leader primers. Otherwise, you do Ig kappa. If Ig kappa is negative, you can use Ig kappa KDE as a surrogate. Otherwise, you can go for Ig lambda rearrangement. So, following this order will maximizes your sensitivity while minimizing the cost. Proposed T cell clinical algorithm is you test for TCRG. To confirm, you can use TCR beta. Only if required, like in immature T cell neoplasms like TLL, TCR-d can be tested for. If TCR-d rearrangement is there and the lineage is assigned, you won't have beta and alpha rearrangement. So that delta deletion which I spoke about will not happen. While interpreting, you will be seeing some pseudoclonal cases and oligoclonal cases. When you give low DNA input, it causes preferential amplification because of the sampling error. You have to repeat the test always. If it is reproducible, that means it is clonal. Pseudoclonality will not be reproducible on repeating. Reactive responses to viruses and due to aging, you can see some oligoclonal peaks like this. So differences between PCR and NGS. The output of PCR is size peak, that is by capillary electrophoresis. It gives you bell-shaped curve, that is because of Gaussian distribution in case of polyclonal population. In monoclonal population, you see sharp spike, which we have already seen in the initial few slides. So PCR can answer only one question: is there any clone? Yes or no, that's it. You cannot determine sequence, you cannot determine somatic hypermutation status. This is exact nucleotide sequence, and the relative abundance also it will tell you. And it will answer your question, who exactly is the clone. If you know the clone, if you know the sequence, it will be easy to track the lineages, and you can measure subclonal diversity, and you can also determine somatic hypermutation levels. Subjectivity which is lying with PCR assays, whereas NGS will give you sequence data. You are seeing the sequence, it will also give you relative abundance, so there is no much subjectivity over there. Background resolution is high in case of NGS. It is limited in with PCRs. You need high quality DNA in both, but some optimal DNA will also work for NGS. Here you will only get the information about the size. Here you get information about sequence. The cost is low, and the test results are rapid in PCR, whereas it is expensive, and the results can be delayed due to the complex workflow. If you are aiming for NGS, it has got high sensitivity. When compared to FR3, FR1 primers have got good sensitivity, but you need a good quality of DNA, because if you design primer at FR1, you will get a good amplicon. For that, you need good DNA. FR3 primers, they work best on degraded DNA, but the sensitivity is low. Always remember the pseudoclonal peaks. You know that it can be used for diagnosis and also for residual disease monitoring. Why this MRD is helpful? Because molecular relapse precedes clinical relapse, so early detection allows early intervention. So to summarize, always try to use screening tests like IgH for B cell and TCR-g for T cell. Just to summarize, IgH rearrangement has happened, then kappa rearrangement. If it is not happening, this Ig kappa should get deleted with the help of kappa deleting element. It will form intron RSS KDE rearrangement here. That can be used as a surrogate to know whether kappa is there or not. Finally, if the IgK rearrangement is not happening, Ig lambda rearrangement will happen. So the kappa deleting element is a crucial diagnostic marker. It rearranges V genes to intron RSS to eliminate non-functional Ig kappa alleles. You don't want to leave any signature behind. That's why you are trying to delete this IgK non-functional alleles. So IgH, because of its universality, the first rearrangement event, it is present in almost all B cells. It has got maximum diversity. So it's a its VDJ junction is a stable, unique fingerprint. So we are targeting IgH. And it is gold standard testing for clonality. So the primary screen always would be with IgH and TCR-g. If it is negative or inconclusive, you go for secondary screen, which includes Ig kappa or TCR-b, depending on your diagnosis. Ig kappa is crucial when somatic hypermutation affects IgH primers. TCR beta, it has got higher specificity. It is used only to resolve equivocal TCR-g results. If Ig kappa is also negative, you can use kappa deleting element as a key target for IgK testing. So the one more question was why TCR-g? Despite being present in minority of T cells, it rearranges early in the early thymocyte development, and it leaves a molecular scar there, like shown here. In the early thymic development, all the T cells will undergo TCR-g rearrangement. If the TCR-g rearrangement is non-functional or unproductive, then only the cells undergo alpha-beta rearrangement. If the TCR-g rearrangement is functional, then this alpha-beta rearrangement will not happen. So this TCR-g rearrangement is the permanent marker which is there on all the T cells. So this biological timing is why TCR-g acts as a universal target for T cell clonality. So either you take gamma delta T cells or alpha beta T cells, you must have TCR-g rearrangement. So it is lineage independent marker, occurs early in ontogeny, covering both lineages, and technically also it is simple, fewer V and J segments. It reduces PCR dropout, and the high detection rate is seen in most T cell lymphomas because of its high sensitivity and simpler structure, as it lacks D segments. Alpha also lacks D segments, but the structure is complex. So the basal clinical algorithm would be screening test IgH with FR1, FR2, FR3 primers. It captures almost 90% of cases. In cases where you feel like somatic hypermutation is there and the IgH assay is failing, you test for IgK and IgK-KD. To further refine in lambda positive tumors, you can go for IgL testing. If you follow this order, it maximizes your sensitivity while minimizing cost. So for screening you use TCR-g. For confirmation you use TCR-beta. Only in immature T cell neoplasms where other receptors have not rearranged, you can use TCR-d. So TCR-g is the workhorse of T cell clonality. So you have to know few traps. If you give low DNA input, normal cells appear clonal due to sampling error. So that is called pseudo-clonality. You have to always repeat the test to see whether the peak which you have got is reproducible or not. If that is not reproducible, it confirms that it is a pseudo-clonal peak. And in aging also, in EBV infections, you see some 2-3 peaks like this, oligoclonal peaks. So this is also not malignancy. You should be very cautious while interpreting the results. So we predominantly use two methods, PCR, fragment analysis, and next generation sequencing. The output of PCR is size peaks, that is by capillary electrophoresis. For normal and reactive population, we see polyclonal bell-shaped curve, which I've shown you in the recent beginning in the presentation. Monoclonal population demonstrates a sharp spike. So what we can answer by doing PCR is that, is there any clone? Is there any a clone which is probably a malignant clone? But we cannot see and determine the sequence of the clone, and we cannot determine somatic hypermutation status by doing simple PCR. We can only say whether the monoclonal population is there or not there. Whereas NGS can answer your question. Who exactly is the clone? What is the exact nucleotide sequence? What is the abundance of that clone in the sample? So it helps in tracking clonotype lineages. It measures subclonal diversity, and it determines somatic hypermutation levels. So to compare, PCR, the traditional method, is subjective. You can call pseudoclonal peak as a clonal peak. You can call oligoclonal peaks as clonal peaks. So there is high subjectivity here. Here the subjectivity is low because your sequencing data is there. You can very well see the sequence information. The background is limited here. The background is high here. You can very well discriminate the background from the true clonal peaks. So background resolution is high, discrimination is good. DNA requirement, you need high quality DNA for PCR. If you take FFP also with shorter amplicons, NGS can work, but still it's a disadvantage only in both the methodologies. Here you get only information about the size. Here you get full sequence. PCR is less expensive when compared to NGS, and the results are fast with PCR. So if you take NGS, it has got high sensitivity. IGH FR1 primers have got sensitivity better than FR3 primers because they cover larger area, but they need good DNA. Whereas FR3 primers, as they cover less area, the sensitivity is low. It works best with degraded DNA. Always remember the pseudoclonality. Low DNA input can mimic a clone. So always correlate with histology. So clonality testing is useful for making diagnosis and also to monitor residual disease. Why? Because molecular relapse precedes clinical relapse. Early detection allows early intervention. So the golden rules: screen first with IGH for B cell, TCR-g for T cell. Watch for mutation at the primer binding sites. Be aware of somatic hypermutations. Verify deletions if your IGH fails and if your IG kappa also fails. Check for IgK and KDE rearrangements and SINT rearrangements, that is constant intron RSS KDE rearrangements. Always the context is king. Peak is a peak without the proper clinico-pathological context. The pitfalls and technical challenges with PCR are DNA quality, allele dropouts, preferential amplifications, pseudoclonalities, and size ambiguity. Different sequences can have same size. It doesn't mean that they are clonal. Sensitivity limits are obviously there, and you may not always get a proper Gaussian distribution for polyclonal neoplasm, polyclonal population, and sharp spike for monoclonal population. So there are, in between there are some ambiguous results which you face issues with. Why one has to move to NGS based clonality? Because it gives sequence level resolution, not just the size. It gives you higher sensitivity and you will be able to detect minor clones, shorter amplicons suitable for degraded DNA. Quantitative clonotype frequencies will be given with the help of NGS data, better comparison across time points and tissues. So advantages we have seen. Limitations are also there with NGS, like it is expensive, complex, you need expertise, you may have data interpretation challenges with FFPE samples specifically, lack of widely accepted standardized protocols and thresholds. So you also need bioinformatics infrastructure and expertise. So clinical applications of clonality testing, as we know that it distinguishes reactive versus neoplastic lymphoid proliferation. You can use it for measurable residual disease. You can always check for relapse versus new clone in recurrence. You can also track clonal evolution and diversity in lymphoid malignancies. Many applications are upcoming, like research and repertoire studies for autoimmunity and immunodeficiency. The challenges and open issues, as I mentioned already, standardization and guidelines for NGS clonality, reporting thresholds are not well established. Distinguishing low-level clones versus background noise, cost, turnaround time, validation in routine diagnostics, low input, degraded samples, integration with mutational profiles, multi-omic assays, regulatory and accreditation requirements, etc. Future directions and innovations: ultra deep sequencing, digital PCR, hybrid methods are coming up, single cell clonality assay which links heavy and light chain simultaneously, liquid biopsy, cell free DNA, circulating clonotype detection, AI machine learning for pattern recognition and interpretation, integrated assays combining clonality, mutation and epigenetics, expanded use beyond hematological and solid tumors. So practical tips for implementation in diagnostic labs: choose primer sets covering multiple loci, validate sensitivity and reproducibility, include appropriate controls and replicates, establish thresholds for calling clonality, cross-disciplinary interpretation should be there, continual quality assurance and proficiency testing to be implemented. So the summary and key take-home messages: clonality testing helps distinguish reactive versus neoplastic lymphoid proliferations. Conventional PCR fragment analysis is robust but has limitations. NGS-based clonality offers deeper sensitivity and sequence resolution. Interpretation must always be in the context. Ongoing advances will push clonality testing into more sensitive and comprehensive use. Thank you.